Today, the class will be on nucleotides. Basically, the topic which I will be discussing will be clinical significance. Actually, without having a basic understanding, it is difficult to understand the clinical impact. And if you know the clinic, if I go directly to the clinic, then there will be requirement to understand and you have to come down back to biochemistry. Without biochemistry, you cannot understand. So regarding nucleotides, you have read in class 12. So many things you know. These need not be noted down and these things are written in the books also. Many things I will tell which you will not get at a time in any book. And some of the pictures which I will show it to you have been taken from our own OPDs. At the end, you will understand that how much you have to read about nucleotides. So, basically, why nucleotides? See, nucleotides are required for all living beings. So, why a medical graduate is required to study nucleotides? So, just like I think we have been covered this carbohydrate, protein. So, basic unit of carbohydrate is a monosaccharide. It's a polymer of monosaccharide. So, basic unit of protein is basically amino acid. Similarly, the basic unit of any nucleic acid is nucleotides. And these are formed, as you know, by the purine and pyrimidine nucleotides only. So, they are the building blocks or precursors of nucleic acid. And DNA and RNA are basically polynucleotides. And this much answer is not obtained from you, my students. Earlier we got what are polynucleotides. That DNA and RNA is the answer, are the answers. That much could not be told before the examiners. So that nucleotide sequence is very much important, as you know, to have the proper protein. If there is any mutation in that sequence, it will form an abnormal protein and it will cause disease. Third is high energy compound. You know about ATP. Not only ATP but GTP also gives an all phosphates in triphosphate form are high energy compounds. They give energy. Adenosine is basically a nucleoside. And during our time, this medicine was not available. Now this drug is used for supraventricular tachycardia. When the heart rate is very high, the heart rate needs to be brought down to normal rhythm, and this drug is used, adenosine, and also as a nuclear stress test as an adjunct. Many a time we cannot do the, all the tests with the conventional tests. So we require nuclear testing. Then synthetic nucleotides, nucleobases, these are extensively used in the treatment of autoimmune diseases, cancers mainly, AIDS, and suppression of that autoimmune phenomena which occurs in our body and for which the disease result. And there is second messenger in the hormone action, that is called signal transduction. When a hormone reaches a cell gets that signal. Hormone is the first signal, and from that arrival of the hormone to the cell surface, the cell membrane transmits a signal, and that is done by cyclic AMP and cyclic GMP that we will read during mechanism of action of hormone action. So coenzymes. So you have heard about coenzymes. The phosphatic group is NAD, NADP, and FAD. You will know a number of reactions catalyzed by NAD and NADP. Biosynthetic intermediates like glycoprotein synthesis, you have seen proteoglycans, glycoproteins. So there UDP glucose and NADP galactose are required. UDP again, it's basically a nucleoside. So similarly, UDP sugar derivative, UDP glucuric acid you will read very specifically during bilirubin metabolism and bilirubin excretion and jaundice. The lipid biosynthesis I think will be covered, and CDP, acyl glycerol will be required. CTP will be required again for the protein synthesis. It is having a diverse load in metabolism, energy metabolism, protein synthesis, a regulation of enzyme activity, and signal transduction, as I have already told. So here you note down very specifically because we have to read the metabolism. So they are regulators of metabolic pathways. Like there are several reactions which are ATP dependent phosphorylations are the key metabolic enzymes of glycolysis, glycogenolisis, glycogenesis. Phosphorylation activates, sometimes phosphorylation deactivates an enzyme. So allosteric regulation, by this time you have read allosteric modification of enzymes. So that makes a conformational changes and the enzyme activity is either increased or decreased. And these are controlled by ATP, ADP, AMP and CTP also. So control of oxidative phosphorylation, this has been also covered, I think. So that is controlled by the ADP. So as principal donors and acceptors of phosphoryl group in metabolism, like ATP and ADP, they give transfer phosphoryl group. And they are the principal players of energy production. Metabolic intermediates, I have already told that UDP glucose methyl group donor. Remember, one important point, yes, adenosine methionine. This is an active methyl donor. And in a short while, I will discuss about adenosine methyl. So, biologically important nucleotides, if you say all the nucleotides basically, except of hybrid derivative, are all important. You may not be knowing about hypoxanthine, but hypoxanthine I will discuss. So hypoxanthine derivatives gives a ionisine monophosphate, ionisine diphosphate, and ionisine triphosphate. These I will discuss in a short while again. Used in DNA diagnostics. It's basically you will be having this in this test PCR because of COVID. Perhaps everybody is aware of RT-PCR, that test. So it detects basically the presence of nucleic acid and DNA. Basically, if there is RNA that is converted to DNA, that is called, that's why in COVID it is called RT-PCR. Basically, it is DNA diagnostic. It's an amplification reaction. And there deoxynucleophosphates are required because at one point of time, whether it is of COVID, whether it is of hepatitis B or it is of human papillomavirus, we don't know. We use a small part that is called oligonucleotide primer, and with that we allow to incubate. If there is presence of that particular DNA, it will bind and it will hybridize. Then that chain length of the DNA polymerization has to be taken place, has to be allowed to take place within the test tube. So that is there. Without this nucleotide triphosphates, chain elongation will not occur. So DNTP is an essential ingredient of all polymerase chain reactions. Now we are having at one, then we require to also terminate. So for that dideoxynucleotide triphosphate, or these are called chain terminating nucleotides. I'll tell you why it is called. Now these DNA PCRs are used in multiple conditions. I have named a few, that is infectious disease, the human immunodeficiency virus. It was first detected long back, then human papillomavirus, mycobacterium tuberculosis, COVID PCR you know, and during cancer typing also. What type of cancer it is, that and what treatment has to be given, for which this PCR, specific PCR is used. The quantify viral load, not only detection, but how much virus is present. That is one of the major criteria of treatment modification in hepatitis B infection. What is the viral load? You have given a drug, and after some time, 2 to 3 months, you want to see whether the viral level has decreased or not. Then the prenatal DNA diagnostic means before the baby is born, whether the baby is genetically abnormal or not. For that you require PCR. It can be done from chorionic villus biopsy. By this time you are aware of what is chorionic villus. Amniocentesis, this Leica amni, that is punctured through abdomen and the fluid is taken, and third is blood. One of my major thrust area to develop a DNA diagnostic center for the Eastern India at Durgapur in 2002 was from blood, because both are invasive procedures. Chorionic villus biopsy, you have to require to take a biopsy from the mother's womb. Amniocentesis, you have to puncture. Why not to do it from blood? Diagnosing genetic disorder like thalassemia, hemophilia, Duchenne muscular dystrophy. Without a confirmed PCR, you will not be able to diagnose. Earlier, the disease was diagnosed based on clinical findings. Now we require PCR, tissue typing, HLA compatibility before organ transplantation. Bone marrow transplantation, HLA haplotyping is done. Forensic, recent case of Arjigor, who is the criminal? That biological material has to be obtained which must contain cells, that is called must DNA. So that evidence must be there, and it is called from any body tissue which is having a nucleated cell. Even a single cell which is possible, then that can be identified whether the person is living, whether the person is dead, or whether it is burnt, partially burnt, or partially putrefied. At least DNA needs to be obtained from that sample. Disputed paternity. It's a big issue for illegitimate child who is the father of the baby. But that has to be identified by PCR only. Adoption. At one point of time, those who are not having children, they adopt a baby, and controversy arises, who will be the legal owner, legal heir. So at that time, that is also whether they are this child, that adopted baby, are the real offspring of their parents, or is the adopted child or not. That it can be determined from DNA testing. That. So, use of synthetic in medical science. Disease, coming to the disease part, cancer, number 1. Then autoimmune diseases, psoriasis, I will show you the picture, viral infection, AIDS, gout. This I will show it once again. This first metatarsus joint is affected. Here, in the acute case, there is an acute inflammation, and if this after this acute phase subsides, this makes a nodule like this. You can see the uric acid deposition here. That causes gout. So this is basically herpes simplex infection. You will see this patient in the OPD, at least in the medical OPD. I have seen once in a week, at least one case in a week. Chicken pox, I think many of you have suffered, have seen. Herpetic keratitis, this I have seen during my house session, that viral infection, and this is staining. Whether you have any doubt, this is the lesion on the cornea, and if you want to confirm it, you stain it with a dye. It will be clearly visible. So for viral infection again, psoriasis, extensive psoriasis of the body. And this patient is from OPD. He attended in the medical OPD last week, and I took the photograph. This was the photograph when the treatment was not started. The patient came for the first time, and after treatment with methotrexate, that is a synthetic analog. This is analog of this nucleotide. I will tell you. You can see the response. These are the most distressful conditions. You can see the cancer mouth, called cavity. Can you see the cancer? Neglected cases. Cancer breast. You see there is axillary load is here. By this time, upper limb, I think we have been told. Metastasis, nipple retraction, metastasis. So stage 4. So now coming to the actual diagnostic part, so all the nucleotides are determined with a single letter abbreviation. Whether it is a free nucleoside or nucleotide, the prefix D is used for DNA, deoxy. ATGC, all you know. The abbreviation of nucleotide by 3 letters. First one is the nitrogen base. Second one is the number of phosphate. Third one is the phosphate, standing for phosphate. So that is ATP, ADP. ATP means adenosine, guanosine, triphosphate, diphosphate. These are the things. So NTP, a nucleotide triphosphate containing ribose. They are the building blocks of RNA. So during polymerase chain reaction, we require DNTP, not NTP. So NTP, what is called nucleoside triphosphate containing ribose. You see ribose, there are 2 hydroxyl groups here, 2 and 3 position. Deoxyribose, this 2 position hydroxyl group is not there. That's why it is called deoxynucleotide. This is deoxyribose, and this is ribose. This is deoxyribose. DDNP, you note down here. This is dideoxynucleotides. Perhaps you have not read it in class 12. So here you see there is a hydroxyl group here, and this hydroxyl group is conspicuously absent here. So there are 2 dideoxy. So normally when a chain elongation occurs during a DNA replication, this hydroxyl group presence is a must. If that hydroxyl group is not present, the chain termination will occur. The chain will no longer be able to extend. Coenzymes. So in adenosine monophosphate, this phosphate group is replaced by, these are the things. One is active methionine called S-adenosyl methionine. Amino acid adenylates, amino acid. Active sulfate, adenosine, this phosphate group is replaced by the sulfate group. NADP, FAD, coenzyme. And many coenzymes and related are derivatives of AMP. As I told, this is one important thing that you may not, perhaps not read. So S-adenosine, 3' phosphate, 5-phosphosulfate. Note down, it is required for the sulfotransferase reactions. It's a sulfate donor. This sulfate group is here. So each sulfate group is transferred during sulfidation reaction. or sulfur conjugates of drugs for excretion. Yes, adenosylmethionine, do not forget that it is a methyl group donor, methyl group transfer, transformation or aminopropylation. So coming to chemistry of pyrimids, nucleotides, I will tell very briefly. You just listen. The structures you note down, and those which you do not know, just note it down, because this video will be available. So purines, you see they are aromatic heterocycles and their numbering you note down. This is clockwise direction and starting from here, N1 to N6. Pyrimidine, it is counterclockwise from this position. So this is the difference between the two. In the opposite direction, this direction is here, up to 6 here, then again it is clockwise. So this numbering is important because we refer bonds with respect to the numbers. So they are having carbons, but in addition they are having nitrogen. And that is the reason they are called heterocycles. So purines, as you know, this is a standard purine ring, and adenine is basically this adenine group here you see, it is replaced by amino group. So at 6th position, it is called 6-aminopurine. Guanine is 2-amino 6-oxypurine. Pyrimidine, this is the structure as I told. So here number 2 position and number 4 position, that is oxygen. So 2,4-dioxypyrimidine. Thymine 2,4-dioxy 5-methylpyrimidine, and cytosine 2-oxy 4-amino pyrimidine. So this is the actual structure. One small mistake is you may commit. Thymine is basically base present in the DNA, but thiamine is a vitamin B complex. It belongs to vitamin B complex. So note down the spelling. This might be one question also MCQ. And during your study of DNA, you will see two things. One is nucleophilic attack and electrophilic attack of the molecules. Just a little bit about what is that thing. So nucleophiles are having excess electrons. Electrophiles are deficient electrons. You note down so that you understand. If you cannot understand while you are reading, nucleophiles, since they are having excess electrons, so they are negatively charged. And nucleophile attack the electrophilic atoms because they are deficient. Attacks the low density electron density because they are having excess electron. Similarly, electrophilic attacks are they are positively charged, they have deficient electrons. So they attack the nucleophilic atoms of a molecule. Attacks with high electron density because deficient electrons is there. Again, this is not known to you. Modification of polynucleotides. See, normally we know cytosine is there, but this is 5-methyl cytosine. At 5 position, the methylation is there. So this is 5-methyl cytosine, and this is 5-hydroxymethyl cytosine. 6 position there is a hydroxyl group. So 5-hydroxy methyl cytosine. So because methyl group is there, methyl group is here also. In addition, there is hydroxyl group. So this 5-methyl cytosine is present in bacterial and human DNA. These are the targets for drug therapy. 7-methyl guanine, 7 position methyl, and 6 dimethyl, 2 methyl groups are there, that is adenine. This structure is important because when we prepare synthetic analog, it must be similar to this structure so that the body or system will not be able to recognize properly. It will camouflage, it will mimic, and then it will act as a drug. So this structure is important for that because you see we are getting infection from bacteria, from virus. So we have to know what are the things the bacteria, what are the biochemical molecules that are unique in bacteria or virus, which is not present in human being. Because if we give the drug, then our body cells will also be affected. So we want to specifically target the bacteria, virus, or infective organisms. So for that you can see this 5-methyl cytosine is present in bacterial and human DNA. But in contrast, 5-hydroxymethyl cytosine is present in bacterial and viral nucleic acid. So drug therapy can be targeted against this because if you can synthetically modify this thing, then this proliferation of bacteria and virus will not occur. So infection will be controlled. 7-methyl guanine and 6-methyl diamine, they are present in the mammalian RNA, and during oligonucleotide chain identification, these are required. And again I am telling you, this methylation is important. Suppose a female child gets to a X allele, one from father, one from mother. Which one is from father, which one is from mother? It is very difficult. You cannot see it by phenotypic appearance. The paternally derived allele is methylated, not the maternally derived allele. So if the disease is from the mother or is from the father, that can be diagnosed from that allele, and from the mothers, this disease can be, that gene can be located. Who is the actual carrier of the disease? So this is two uncommon things, that is hypoxanthine and xanthine. The uncommon bases, and this exists in lactin form, that is, it is a keto form or enol form. This is the keto form. This is the keto form, and this is the OH form. This is ketoenol form. They can exist in both the forms. Uric acid. This is uric acid structure basically. Hypoxanthine, xanthine, and from hypoxanthine, xanthine, uric acid is produced. So they are also minor bases. So notable purines are basically hypoxanthine, adenine, guanine, that you know, hypoxanthine, xanthine, and uric acid. There are others also, that is theophylline, theobromine, caffeine. So these you may not remember, only remember at least these 5. So like purine, there is again modification of pyrimidine bases. Like here, there at 5 position there is methyl group. So this is 5-methyl cytosine, and there are 2 oxygen here. So this is called dihydrouracil. So sugar group, as I told in the beginning, that ribose is having 2 hydroxyl groups. A 2 position hydroxyl group is there, but in deoxy it is not present. That's why it is called 2-deoxy. And the numbering of ribose is done with a prime. You see the numbering, if you have noted that numbering of purine and pyrimidine, we have noted with only numeral. The number of carbon atoms in pentose sugar is done with a prime sign, only to differentiate which one is from purine or pyrimidine or which one is from sugar. Because in subsequent slides you will see that we denote everything with respect to ribose sugar, that is 3 prime, 5 prime, 5 prime, 3 prime. So what are the roles of different carbon sugars? the roles of carbon in the sugar, pellucid sugar. So first carbon ribose or deoxyribose, it is attached to the purine base or pyrimidine base. Second, this differentiate deoxy and oxy. Third one basically is involved in the phosphodiester bond. Third one. Fourth one basically maintains the structural conformity. The structure is maintained by fourth, and fifth one is meant for phosphate group attachment. So these 3 and 5 is the most important point. 3 prime is basically for the phosphodiester bond. At 5 position, phosphate group is attached. At 3 position also phosphate group can be attached. I can show it. So nucleotides are basically N glycosides. Why N glycosides? Because N is here. We don't have a CC glycosides only in pseudouracil, that you will see. So N glycosides, so N of 1 and C of sugar is attached in purine. N, C, it is the number numeral without prime, the number is here with prime. In pyrimidine, it is 1, 1. So base plus sugar is nucleoside, that you know. But this part also you know, adenine plus ribose, adenosine, guanosine, cytosine, cytidine, remember. And this uracil is uridine, hypoxanthine is ionosine, xanthine is xanthosine. And deoxyribose all you know. Only here new things, this hypoxanthine and xanthine part. So base plus sugar is nucleoside. So nucleotides, if you add phosphate to nucleotide, then it becomes nucleotide. So they are phosphorylated nucleosides. So we have seen this thing, and in addition we have attached phosphate group. You see phosphate group is here. This you have seen. I told you one thing that about pseudouridine. Normally it is an N-glycosidic bond. So this is N-glycoside, that is uridine. You see N-glycoside. Now there is here, it is in number 1 position. Now this molecule rotates. And you see the position number changing from 1 to 1 here, 2, 3, 4, 5. Now 5 and 1. This is basically CC glycosidic bond, not N-glycosidic bond, CN glycosidic bond. That is the difference between the uracil and pseudouracil. And this is important. You can see this structure that I will discuss in the next class. There is presence of pseudouridine in one arm of T arm, that is pseudouridine here. So this CC bond allows more flexibility and conformational flexibility and rotational freedom. And again in this pseudouridine there is extra hydrogen here, which can act as a hydrogen donor also. So this is the uniqueness of pseudouridine. So 5' nucleotides are often seen because this 5' phosphate is attached to 5', but it is written without prefix normally. So 5' AMP is abbreviated as AMP only. So base plus sugar plus phosphate is nucleotide, and specifically you have to note down here, that is uridine monophosphate or uridylic acid. Ionosine is ionosine monophosphate or IMP or inosinic acid. Rest you know AMP, GMP, CMP, and again DMP, DCMP. So similarly, just like uridine and pseudouridine, there will be uridylic acid and pseudouridylic acid. Again, you see the bond is here between N1 and 1, and this is 5 and 1, that is CC bond. This 5 prime, this phosphate, as I told you, normally it occurs in the 5 prime position. Normally it occurs, but at 3 prime position is also there. So 3 prime variety is there also, phosphate group. So 3 prime variety is always written as 3 prime AMP. There is a difference between the two. When you write AMP, means it is 5 prime. But when it is 3 prime, you have to specifically indicate that it is 3 prime. Because normally it doesn't occur. So to specify that thing. So these two adjacent nucleotides are held together by phosphodiester bond. This is the phosphodiester bond. This is the ester bond. So why 2 diester? Where is the 2 diester? One is formed at the time of nucleotide formation. Remember what are the 2 ester bonds in phosphodiester? As I told you here, there is a common picture you will see. The 2 nucleotides, adjoining nucleotides, are joined by a phosphodiester bond. But here, one nucleotide is joining with another, with the phosphate group. So that's why I am telling you, at the time of formation of nucleotide, there is one phosphate group. There is one ester group here. Because here, this phosphoric acid is attached to this 5 at 5 position, so one water is removed. So one ester bond is formed here. This is number 1 ester. And during subsequent addition, the second ester is here. So that is the reason why it is called 2 ester, diester. So 2 adjacent nucleotides are joined together via phosphate to 2 diester bond. So 2 diester bond, phosphodiester bond is connecting basically 2 adjacent nucleotides, and this is a very strong bond and it can withstand temperature. So this part perhaps may not be required written in your book. There is thermodynamic stability of the phosphodiester bond. So normally this phosphodiester bond is cleaved by phosphodiesterase enzyme. So it is such a stable bond that even in the absence of that normal enzymatic catalysis, it is extraordinarily stable and it can live with a half-life of nearly 1 lakh years. Actually, I attended one conference at Center for Cellular and Molecular Biology at Hyderabad, in 2008. A Japanese scientist was trying to do an experiment from a dead rat which was kept frozen in the laboratory. He took the cells from that rat, identified the DNA, and then put it in the egg cell. We wanted to see whether the cells can replicate. And for long 10 years, he could not succeed. At one point of time, after 10 years, he found that cells started replicating. That means the extinct animal, if their DNA is really present, they can be grown into the proper cell culture and DNA study can be done from that. So that was the basic objective of that Japanese scientist. So 10 long years it failed. So because that phosphodiester bond is very stable and DNA was present in the dead mouse also. So in all biochemical reactions there are 2 things. One is enthalpy, the total heat of the system, and entropy, the randomness of the molecule. These determine the rate of reaction, whether it will be a spontaneous reaction, whether it will be a catalyzed reaction, whether the reaction will be favored in this side or that side. that depends on the laws of thermodynamics, and the three laws, and the entropy and enthalpy of the system, and most of the biochemical reactions are actually dependent on the entropy or randomness of the molecule. So as you can understand why DNA is detected in the fossils, because it can withstand that the phosphodiester bond is very much stable, and that's why in the absence of phosphodiester enzyme digestion, it can persist over a considerable period of time. RNAs are less stable. So in ATP, which we have already aware of, universal energy currency. In ATP, there are only two high energy phosphate bond, and this is alpha bond, this is second phosphate, this is beta, and this is gamma. So these phosphate is normally present in everything. That doesn't give a high energy phosphate. Only this second and third, that is beta and gamma, they are high energy phosphate bonds, and they give 7.6 calorie energy per bond. And they are required for endergonic reaction. What are endergonic reactions? Those chemical synthesis which will require energy. And by this time we are aware that some chemical reaction has occurred where ATP was used or ADP was used. That energy was obtained from that high energy phosphate bond. And during muscle contraction, nerve impulse conduction, transport, this energy is required. Because in our body, while we are sleeping, the intestine is moving. Brain cells are also working. So where from they will get the energy? So normally cyclization of the AP normal structure you have seen. Now there is cyclization between 3 and 5. Phosphate is here. So this is called 3' 5' cyclic AMP. Similarly, it can happen in GMP also. So this is the reason I am telling you. I told you that always we refer with the 3' 5' to indicate the ribose, not the pyrimidine that is numbered without having any prime number. So this is 3' 5' cyclic AMP or cyclic AMP. It is a very important compound. And the phosphodiester bond is between the 3' and 5' position of the ribose group. And cyclic AMP and cyclic GMP are the metabolic regulators, and they are second messengers also. So here one thing, the mean intercellular concentration you may not be doing, that is 1 millimole per liter. In contrast, this intercellular concentration of cyclic AMP and cyclic GMP is 6 or less, because they are not required that much amount. ATP is required, but cyclic GMP is required only for metabolic regulation part and for signal transduction. Important characteristic point of nucleotides, they can absorb UV light at 260 nanometer wavelength. So if the human body is exposed to UV lights for a prolonged period, there will be absorbance by the nucleic acid, and that will form thymine dimer. So UV light induced thymine dimers will be formed, and that will not allow the chain elongation or proper DNA replication. There will be mutagenesis, that will be cancer, or there will be cell death. So human bodies are not allowed to exposed in any kind of radiation, including UV radiation. UV radiation is normally required during experimental procedures in the laboratory. So here in this part I have discussed is 3' 5'. So this 3' 5' phosphodiester bond is here. You see, this is directional. When nucleotide synthesis replication occurs, either one is, whether it is from 3' to 5' or from 5' to 3' end. So it determines whether which direction. Normally the sequence representation is 5' end on the left side and 3' end on the right side. So here you see 5' end. So the bond is here, is 3' and 5'. So when the nucleoside change elongation will occur, basically the bond will occur between 3' to 5'. Somebody can say also 5' to 3', but in essence the 3 is on the front in the first position and 5 is on the second position. So normally chain elongation occurs in the 5' to 3' direction, always. But this phosphodiester bond is here. Remember, always the sequence representation is in this form only. But this phosphorylation is with the 3' and 5'. A synthetic analog coming to the end of this class. That pictures you have seen, that cancer pictures, these viral infections. Now you will be able to treat. This chemistry part is very much important to have a synthetic analog, a substance which is basically similar but not identical. There is some difference in the molecular structure or atomic. So it is a structural analog. It's structurally similar but slightly different in composition. Either there is a replacement of atom or a functional group. So that the atom is not absolutely identical. There are two different things. Identical means exact copy. Photocopying a paper, you are getting an identical copy. But similar things, you can note it down, you can have it in similar things, same things, or you can type it down, something like that. Similar and identical, try to understand the difference between the two. So they act by molecular mimicry because they camouflage the system. That small changes, the body may not be able to recognize, or that virus or bacteria, and that deficiency is taken care of. Because we have to design a drug which will not at least destroy our own body cells. And because of the process, it is not absolutely perfect. So during the cancer treatment of growing cells, the growing cells of our body, that is bone marrow, gonads, skins, epithelial cells, which are also proliferating at a higher rate, they get involved. And when the cancer patients are treated, you have seen perhaps one or two cases there is alopecia, the hairs are removed, the skin ulcers are there, and there is a bone marrow suppression. There is the cells, formed cells, RBC, WBC, platelets. They are also getting proliferated in huge numbers. So the moment you give an anti-cancer drug, so that drug will diminish the growth of the cancer cell. At the same time, it will stop the growth of the normal cells. So every attempt is made so that the human body cells are not affected. Specifically, we are targeting the cancer cells, and that cancer targeting is done by detection, is done by the DNA PCR, which type of cancer it is and which drug will be basically required, so that the cancer cells specifically get affected, not the human body cells. So mechanism of action, as I told you, they are similar in structure. They are competitive inhibitors. Enzymes, you have seen the competitive inhibitors. Enzymes have been taught. You know the competitive inhibition. All of you are aware of competitive inhibition. If you don't know, then you cannot understand. See, if something is present in higher concentration, so that will try to overpower the other components. So in body, we are having all these compounds, that DNTPs, but these are also DNTPs. They are similar but not identical. So if they are given in high concentrations, so they will compete with the natural substrate. So they will be incorporated in the DNA. And so the functionally defective DNA will be produced, and it will cause either defective DNA formation, and that will cause arrest of cell cycle, called cell death also, and that is what is desired in cancer. Or highly proliferative disease, where cellular proliferations are very high. So this will be very easier for you to understand. So this drug is given for gout. Gout basically occurs due to high uric acid level. And uric acid is the end product of urine catabolism. From purine, at the last stage we get hypoxanthine to xanthine to uric acid, as this reaction is catalyzed by xanthine oxidase. Now this drug allopurinol is similar to hypoxanthine. So if you give allopurinol, allopurinol will compete with hypoxanthine and it will not allow uric acid to form. It is a structural analog of hypoxanthine. So if we treat this patient with allopurinol, so uric acid production will be decreased and this disease will be cured. In the acute stage, you can see the uric acid crystal, but in the normality, the acute stage, it was recommended not to be given, but at a later stage, after the infection has been controlled. Psoriasis, this patient I have seen last week, as I told you, it attended, this is our hospital chair. This old photograph, the methotrexate was given. So methotrexate, what is methotrexate? It's a folic acid antagonist. Here it is having a structural similarity. So it is used in the treatment of psoriasis, autoimmune disease, breast cancer, lung cancer, in choriocarcinoma. So they are structural inhibitors of folic acid and it inhibits dihydrofolate reductase. Note down this thing. Dihydrofolate reductase, and you read this cycle, folate trap. This is a very short note, common short note during examination, or this may be asked. Specifically, you remember the methotrexate is a structural analog of folic acid and it inhibits dihydrofolate reductase. And this is used in the treatment of autoimmune disease mainly. Now, normally nowadays it is given, but it was also used for cancer. So it is an anti-cancer drug. So if somebody is given a methotrexate, don't jump that you are given an anti-cancer drug. These are given for immunosuppressant agent. So in psoriasis it is given for immunosuppressant because psoriasis is essential and autoimmune disease. Herpes simplex, the moment you see, once you see a single case, you will be able to diagnose. So this is the guanosine structure, and a similar structural analog is formed, that is called acyclovir. And acyclovir is given during the treatment of chickenpox or herpes simplex infection. So they will compete with this is present in the viral DNA. So viral DNA synthesis will be stopped. So this will be cured. Normally chickenpox, there is no treatment was required during our time, but since people are very much aware, so now we give acyclovir to those who are suffering from chickenpox to have a faster recovery. But herpes simplex will not go so easily, so acyclovir is required. Cancer, this is the most distressful treatment. You will see cancer patient, you will treat the patient, but to be very frank, at the end, the extensive amount of money is done, but hardly we get cure if the disease is at the stage 4 stage. So there the treatment, you see purine structure is here. So a structure similar, there is a modification. So thioguanine and mercaptopurine. So mercaptopurine is given for acute lymphoblastic leukemia. This is cancer. Azathioprine is also an anti-cancer drug. So these are nucleobased. There is purine analog. Purine means this is purine structure. So this is a purine analog, mercaptopurine and thioguanine. So similarly, guanine, 8-azoguanine, thioguanine. This is nucleobased purine analog. This is pyrimidine analog, uracil and 5-fluorouracil. At 5%, as I told you, there is only replacement of either an atom or a functional group. But otherwise the structure is same. So the body will take up this 5-fluorouracil and it is used in the treatment of cancer, stomach, colon, rectal, breast cancer. So there are a number of anti-cancer drugs which are given depending on the stage, depending on the case. So this is nucleobased analog. The treatment of HIV infection, this is nucleoside. Pyrimidine is a nucleoside. Phosphate is attached here. You see there is a structure here. This structure this is the difference. Can you see? So digilovulin is used in the treatment of HIV infection or AIDS. So this is nucleoside analog. Acute lymphoma, this is cytidine. Again, this is a nucleotide 2' deoxy. This is, you can see this structure, and this minor change has occurred here. So that is cytarabine. That is again given in the case of acute myelodysplastic leukemia. So this is nucleoside analog. So purine analog you have seen, nucleoside analog you have seen. So there are other nucleoside analogs. So you need not remember. So anti-cancer, number of anti-cancer drugs are there. So in which cancer you will select which drug, that depends on the stage, cancer and type and general condition of the patient also. So I think time is up and you will be able to at least describe the nucleotides and its clinical significance. Is it okay? Now you just read from the books so that you know the basic part, treatment part, analogous, structural analog part you will know in detail during pharmacology, and when you will be in clinical practice, you will get the real test of what you have learned in biochemistry. Okay? Thank you.